In this video, I'll be talking about pancreas. So on pancreas, I'll be making 4 videos. This particular video will be about glucagon. Then another one will be on insulin. Then the third one will be a comparison between insulin and glucagon. And then I'll make another video on the diseases of pancreas. So to start with, let's first talk about the anatomy of pancreas. So in anatomy, what do you have? Like this is a diagram of a pancreas. Okay. Now pancreas, they are elongated and they are tapered organ. Okay, which is also an endocrine gland. It has also has got some exocrine parts. So this is a diagram of a pancreas. Okay, so out here, this entire portion is the pancreas. Okay, so this is the pancreas body. It consists of a tail, and there's a head, there's a pancreatic duct, and this pancreatic duct it actually carries all the pancreatic juices, that is the enzymatic juices and pancreatic juices that comes out and which helps in the process of digestion. Okay, all those things will come out of here. And the other part is the endocrine part, which consists only 1%. Now, if we get a section of a pancreas, it almost looks like this, okay? So we have this pancreatic acini, which is the exocrine part. Okay, it's the exocrine part. That means from here the enzymes, that is the digestive juices and everything will come out. And the endocrine part is the islets of Langerhans. Now, within islets of Langerhans, we have alpha cells, we have beta cells, and we have delta cells. Okay? Now, if we uh, see the arrangement, we can see that the cell, like within the islets of Langerhans, this is one islet of Langerhans. Now, within this islet of Langerhans, we can see the alpha cells are present towards the periphery, okay, and the beta cells are present on the inside. Why? Because beta cell produces insulin, and insulin is a very, very important hormone. So it's present on the inside. And the third one is that the last type is the delta type. Now delta type of cells, it's spread out, like it can be found inside, it can be found outside, wherever. Okay. So basically we have alpha cells, we have beta cells, and we have delta cells inside the islets of Langerhans. Okay. Now the alpha cells produces glucagon. Okay. How do you remember? Glucagon A, alpha A. So from there you can remember. Now for beta cells, it produces insulin, and there is the delta cell. This one produces somatostatin. Okay. All right. Now, what is the function of glucagon? The function of glucagon is to produce glucose. Okay. The function of insulin is to take up the glucose from the blood and to convert them into glycogen. And somatostatin has an inhibitory function to both glucagon and insulin. Okay. Now, let's move on to the endocrine part. Okay, so our pancreas is actually an heterocrine organ. Now, what do you mean by that? Heterocrine means it has one part is exocrine and the other part is endocrine. Okay, now within the exocrine we have pancreatic acini, which consists of 99% of the pancreas, and endocrine is islets of Langerhans, which is only 1% of the pancreas. Okay, so from pancreatic acini, pancreatic juices are produced, digestive enzymes, for example, lipase, trypsinogen, etc. And from the islets of Langerhans, we get alpha cells, beta cells, and delta cells, from where we get glucagon, insulin, and somatostatin. Now let's talk about when glucagon is secreted. Glucagon is secreted during hypoglycemia. which means that the concentration of glucose in the blood is less than 70 mg per dl. That is the concentration of glucose within the blood. Okay, so that means I'm talking about blood glucose. When the blood glucose is less than 70 mg per dl, then we are suffering from hypoglycemia. In that condition, glucagon will be secreted from the alpha cells. Okay, now in case of insulin, insulin is secreted when there is hyperglycemia. That means when the concentration of glucose, blood glucose, is more than 130 mg per dl. So the question is, what is the normal range? The normal range that we have is 80 to 120 mg per dl in the blood. Okay, this is the normal range. Normal range of blood glucose. So this is the normal range 80 to 120. Now whenever the blood glucose is low, okay. So whenever the concentration of the blood glucose is low, in that time glucagon will be secreted, alright. And whenever the concentration of the glucose is more than 120, that is if it reaches more than 130, then what will happen? Insulin will be secreted. Okay, that will give a signal to the insulin. That's how glucose and insulin are secreted. And somatostatin is a secretion which actually inhibits both insulin and glucagon. All right, so next we will discuss about the production of glucagon and how they are produced. Okay, and then we'll move on to its function. Okay, so out here we are in a fasting state. That means we are not eating enough food. So in that stage, what will happen? In that particular stage, the amount of glucose in our blood, so the blood glucose will be the concentration of the blood glucose will be low. Okay, so in this condition, when the blood glucose state is low because we are in the fasting state, so what will happen? Very less amount of glucose will enter inside the alpha cell. That means this is the alpha cell, means it's a cell of pancreas. Okay, so that means in the pancreas, very less amount of glucose will enter. So the amount of glucose that is entering is very low. All right. So now what happened? This glucose will now get converted into pyruvate. Now, since the amount of glucose is low, the amount of pyruvate will also be low. Then the pyruvate will enter inside the mitochondria, will go through the Krebs cycle. And from here, ATP will be produced. And the amount of ATP produced will also be low. Now this ATP will come and bind to this receptor. It's an ATP receptor, okay? And because it's binding, and again remember the amount of ATP that we have in our body is low because the amount of glucose in our body is also low. So everything is low, okay? So in that case, very less amount of ATP will be produced, and this ATP will go and bind with the receptor, okay? Now because of this, what will happen is there will be an accumulation of potassium ion within the cell. So there will be an accumulation of K plus, and because of this accumulation, what will happen? The membrane depolarization will decrease. Membrane depolarization will decrease. Now, because of this, what will happen? Since the concentration is low, therefore, this low membrane depolarization will keep this open. Now, what is this? This thing is actually a calcium channel. So because of this particular action, what will happen? The calcium channel will remain open. Okay, so now since the calcium channel is open, what will happen? Calcium will enter inside, obviously. So from outside, calcium is entering. Okay, calcium from outside is entering inside the cell. Now let's see what else is happening inside the cell. Now this cell also has got other GPCR protein. Okay, they have other GPCR proteins as well. Now on this GPCR protein, some other ligands will go, it will come from the blood and come and attach out here. Okay, for example, secretin and all. Okay, this will come and sit. It can be either norepinephrine or it can be epinephrine. All these things will come and bind out here, okay? And this will give a positive signal to the nucleus. Now inside the nucleus there is a DNA present. Now from this DNA, an mRNA will be produced. Then this mRNA will come out, okay? This is the rough endoplasmic reticulum. Okay, from this in this rough endoplasmic reticulum, a vesicle will be formed, okay, and modification and all of that will take place. Then this vesicle will come out and it will reach the Golgi apparatus, where again the vesicles will be formed and the vesicles will be released out here, okay, within the cell. Now this vesicle, it actually contains what? It contains glucagon. So the gene for the production of glucagon is present within the alpha cell, right? So this is how the alpha cell is getting signal and the glucagon is produced. Now in order for this glucagon to fuse with this membrane, it requires calcium. So the calcium will come out here and it will help, and therefore the cell, not the cell, but the vesicle will fuse, and therefore the glucagon will come out. Okay, so this is how glucagon is released. So this is glucagon. Okay, so this is how glucagon is produced by the alpha cells. Then what will happen? This glucagon will go into the bloodstream. So that means whenever we are in the fasting stage and there is low blood glucose, our body will be producing glucagon. Okay, so the production of glucagon has been done. Now we will see what is the function of glucagon and how it's going to function. Okay, so first let us come to the liver cell. Okay, in the liver cell, what will happen? This glucagon will come and this glucagon will come out here and will sit out here. Okay, with the receptor. So this is glucagon. Okay, glucagon came and sat on the receptor, and because of which what will happen? This is a GPCR. It is bound to a G protein, which is bound to a GTP. So now what will happen? This will go and activate the effector enzyme adrenal cyclase. Now what will happen? Adrenal cyclase will convert ATP into cyclic AMP. Then this cyclic AMP will then help in the production of protein kinase A. Now, within the liver cell, we have a storage of glucagon. Now, what will happen? This glucagon, now again, remember why we are actually producing glucagon? We are producing glucagon so that the amount of glucose in our blood can be increased, because remember, glucagon is only secreted when the blood glucose level is low, and also all these things is happening during the fasting stage when the blood glucose level is below 80. Okay, that means, or you can say below 70, because till 70 it's quite normal, but after 70 it starts giving problems, so we start producing glucagon. So now what will happen within the cell, since the glucagon is there and it's giving all the signals, so this pKa will give signal, and therefore this glycogen will now be converted into glucose. This is what we are trying to get. Okay, we are trying to get glucose. So this glycogen will be converted into glucose. Now what is glycogen? Glycogen is the stored form of glucose, right? So this is how the glucose is stored in the form of glycogen. It's a very big molecule. So this glycogen will be broken down into glucose. Now this particular process is called as glycolysis. So this is called as glycolysis. That is the breakdown of glycogen is glycolysis. Okay, so by the process of glycolysis, glycogen is broken down into glucose. All right, now what else? Now what will happen? This glucose will then go back to the blood, okay, increasing the concentration of glucose, which is what we are trying to do. But just breaking down glycogen alone is not going to help. We need to do some more things. Okay, so let's see where else our glycogen is going to work. So again, we have this glycogen in the blood. It will go and work on this. Okay, it will go out here. What is this? These yellow cells are adipocytes. That means the fat cells. Okay, adipocytes or fat cells. Okay, now within the adipocytes, again we have a GPCR, G protein bound to GTP, which will go and activate the effector protein adenyl cyclase, which will convert ATP to cyclic AMP, which will activate protein kinase A. I think you all know all of these, right? So, okay, so it will give a positive signal to what? We have triglycerides present out here, big molecule, okay, 3 long fatty acid chain and a glycerol. So what will happen? This will break down this particular molecule, okay, all the triglycerides with 3 fatty acid chain and 1 glycerol within the fatty acid or within the fat cells will be broken down, and when they are broken down, they'll be broken down into what? They'll be broken down into glycerol, and they'll be broken down into odd chain fatty acid. Okay, now this glycerol and fatty acid, they will go into the bloodstream. Okay, so here we have the glycerol. Okay. So there you have glycerol, and we have this 3 fatty acid chains. Now what? This glycerol fatty acid chain will again enter inside the liver cell. Okay, so entering. This is the glycerol, and the fatty acid chain is also there. What kind? Odd chain fatty acid is also entering. Okay. Now, now what happens is this glycerol is again broken down into glucose. The fatty acid chain is also taken up and is converted into glucose. Also, there is amino acids present out here, which is also converted into glucose. Now, all these processes are new processes which is taking place. That means new glucose have been made. So, this particular process is sorry, gluconeogenesis is taking place. This four, okay. Gluconeo means new. So, new glucose have been made. Okay, not this one. Okay, so new glucose are being made. So this process is called as gluconeogenesis. And whatever glucose has been made, that glucose will now, this glucose will now come into the blood. All right, and therefore increasing the concentration of glucose of the blood. Now, when the concentration of glucose in the blood increases, then what will happen? When the blood glucose level increases, it has to increase up to 80 to 120 mg per dl, which is the normal range. When it reaches till that, then the production of the glucose will stop. Okay, so out here, the process that is taking place out here where the glucose, glycogen, and the orange and fatty acid is produced, this process, this is called as lipolysis. Okay, so lipolysis is also done by glycogen. Okay, now this one. So now heart cell. So what happens in case of a heart cell? So I have made a heart cell now within the heart cell because of the presence of glucagon. So then a glucagon will come and bind out here. Glucagon. Okay. Glucagon comes and binds out here. So what happens is again a G stimulatory protein will be activated. Okay. Then which is more binds to GTP. So it gets active. Then it activates a affected enzyme adenylyl cyclase, which will convert ATP into cyclic AMP, which will activate protein kinase A. Okay. Then this protein kinase A will give positive signal to all these channels. Now, what are these channels? These channels are all calcium channels. Okay. So the calcium channels will open up. Okay. Now, because of the opening of the calcium channels, what will happen within the blood cell? Sorry, heart cell. They will have higher contractility. So that means whenever there is low glucose in the blood, whatever glucose is not present, that has to be reached to each and every part of the body. So therefore, the heart will start beating very very fast. Okay, so that like the blood can reach every and each and every part of the body, and there is at least they are getting whatever glucose is present within the blood for that amount of time is being given to each and every cell of the body. So otherwise, it can have very very grave consequences. The cell might even die. So therefore, uh, the heart will start beating very fast because of the presence of glucagon in the blood. Okay? So these are the major functions of glucagon. Next, I will discuss about the functions of insulin. Okay, then I'll make a comparison between glucagon and insulin once again.